The AtezoTRIBE study is a phase 2 randomized trials aiming at evaluating the efficacy of the anti-PD-L1 atezolizumab when added to an upfront treatment of FOLFOXIRI plus bevacizumab in unresectable metastatic colorectal cancer patients. FOLFOXIRI bevacizumab is now recognized as a valuable upfront treatment option, at least for a subgroup of selected metastatic colorectal cancer patients. Based on the results of a recent individual patient database meta-analysis, including 5 randomized trials, FOLFOXIRI bevacizumab demonstrated a significant advantage in terms of survival, also at long term, when compared with doublets plus bevacizumab. And the patients with right-sided tumors and those with RAS mutant tumors appear as the best candidates to receive this upfront intensified treatment if they are in good general conditions and not too old to receive such an intensified regimen. Now, checkpoint inhibitors have definitely provided amazing results in microsatellite unstable metastatic colorectal cancer patients that are around the 5% of all our metastatic colorectal cancer patients, but failed to demonstrate a significant clinically relevant efficacy in microsatellite stable tumors, which are the vast majority of metastatic colorectal cancers. And there is a strong biologic rationale for combining a checkpoint inhibitor with the anti-angiogenic bevacizumab in order to make a cold tumor, such as microsatellite stable colorectal cancer, to turn this cold microenvironment into a hotter microenvironment, because bevacizumab is able to potentiate the function of dendritic cells. It may increase the infiltration of T lymphocytes, cytotoxic lymphocytes, and it may also decrease the efficacy of some immunomodulatory cells, such as lymphocytes Treg. So this is the rationale for combining the checkpoint inhibitor atezolizumab with the anti-angiogenic agent bevacizumab with a more intensified upfront regimen of chemotherapy, which is able to release a high amount of neoantigens. The idea is that when we release a high neoantigen load into the tumor, then these immunomodulatory properties of bevacizumab may act together with the checkpoint inhibitor in potentiating the anti-tumor activity. AtezoTRIBE is a phase 2 randomized trial. The primary endpoint is progression-free survival, and we aimed at finding out an advantage in favor of the addition of atezolizumab versus the control arm, with a null hypothesis of 12 months of median progression-free survival in the control arm. We aimed at finding out an hazard ratio of 0.66 in favor of the experimental arm, with alpha and beta error of 0.1, one side alpha, and a beta error of 0.15. So these parameters are not so strict as in the context of a phase 2 comparative randomized trial. So this is not a phase 3 comparative registrative trial, but a proof of concept to find out the potential usefulness of this therapeutic strategy. 201 patients had to be randomized. There were no molecular criterion of selection, so both MSS and MSI high patients with MSS and MSI high tumors were eligible for this trial, because when the trial was conceived, we had not available such a strong evidence of efficacy of checkpoint inhibitors in macrosatellite unstable tumors. And in around 20 oncology units in Italy, the GONO Foundation conducted this independent no-profit trial randomizing metastatic colorectal colorectal cancer patients deemed fit to receive an upfront intensified combination regimen with FOLFOXIRI bevacizumab in a 2-to-1 ratio to add atezolizumab to this treatment. The intensified combination regimen, either FOLFOXIRI bev in the control arm or FOLFOXIRI bev atezolizumab in the experimental arm, were administered up to 8 cycles, then followed by maintenance with F5FU and bevacizumab with or without atezolizumab according to the randomization arm until the evidence of disease progression or unacceptable toxicity or consent withdrawal. And after the evidence of disease progression, the reintroduction of the same regimen used up front was recommended as per protocol. The trial had an initial safety run-in phase to rule out any special issues in terms of safety, so that the first 6 patients were treated only at the coordinating centers, and then all the other satellite centers were open to the enrollment in the trial. And the accrual rate was really very fast as a consequence of the high interest of the scientific community in making immunotherapy a useful option also for microsatellite stable patients. 218 patients were randomized. These patients were, in the vast majority of cases, in very good general conditions with the NICOG performance status of 0. The median age was 61 years, and most of patients, more than 80%, had synchronous disease. Only the 27% of patients had a liver-limited disease, and around half of included patients had a right-sided primary tumor. Overall, around the 80% of included patients had a right-sided and or a RAS mutant tumors. So they were the so-called potentially best candidates to receive an upfront treatment with FOLFOXIRI plus bevacizumab. And the 6% of patients included in this trial had a microsatellite unstable tumor. All the others had microsatellite stable metastatic colorectal cancer. We had no special findings in terms of safety. Chemotherapy-related adverse events occurred within the expected frequency, both in the control and in the experimental group. So no added toxicity in terms of chemotherapy-related and bevacizumab-related adverse events when adding atezolizumab to FOLFOXIRI plus bevacizumab. And also no special findings in terms of potentially immuno-related adverse events that occurred within the expected frequency. With regard to the primary endpoint of the study, progression-free survival, the addition of atezolizumab to FOLFOXIRI bevacizumab was associated with a significant benefit in terms of progression-free survival, with a median PFS of 13.1 months in the experimental arm as compared with 11.5 months in the control arm, a hazard ratio of 0.69, which is statistically significant. When looking at the response rate and the R0 resection rate, no difference was observed between the two study arms. When looking at the subgroup analysis in terms of progression-free survival, according to the most relevant clinical and molecular characteristics, a significant interaction effect between treatment arm and microsatellite instability was found. In particular, those patients with deficient mismatch repair, and this evaluation was performed locally through immunohistochemistry, these patients with dMMR tumors had a huge benefit from the addition of atezolizumab, somehow as expected, and at a median follow-up of around 21 months, the median progression-free survival was still not reached among the small subgroup of microsatellite unstable tumors treated in the experimental arm. Among these 8 patients versus 5 in the control arm, there was an hazard ratio of 0.11 in favor of the addition of atezolizumab to FOLFOXIRI plus bev. But also when looking at the larger subgroup of patients with microsatellite stable tumors, however, there was a strong signal of efficacy when adding atezolizumab to FOLFOXIRI plus bevacizumab. The extent, the magnitude of this benefit is less impressive than compared with DMMR tumors, but there was a significant benefit also in this subgroup. So that if we had conducted the study only in the subgroup of MSS tumors, still the primary endpoint would have been met, since an advantage from the addition of atezolizumab was found out, 12.9 median progression-free survival versus 11.4 in the control group among MSS patients, with an hazard ratio of 0.78 and a p-value of 0.071, which is significant according to the initial design of the study and the one-sided alpha error of 0.10. So also in the subgroup of patients with MSS metastatic colorectal cancer, there is a signal of efficacy in terms of progression-free survival when adding atezolizumab to upfront FOLFOXIRI plus bevacizumab. As previously mentioned, the AtezoTRIBE study was a phase 2 randomized trial, so I think it's good food for thoughts and for future research experiences in this field. We have now ongoing a large program of translational analysis aiming at investigating whether we can identify some biomarkers, predictors of benefit from the addition of atezolizumab in the microsatellite stable subgroup. So I believe that this is not a practice-changing study immediately tomorrow, but I definitely believe that this strategy may be a good starting point for future investigations in this field. At the same time, in the meanwhile, I think that some other hypothesis generating data will come out from this trial, thanks to the evaluation of some candidate biomarkers, including the immunoscore, signatures, predictive mRNA signatures, potentially predictive of benefit from checkpoint inhibitors such as the TERMAIO, but also the tumor mutational burden, the infiltration of T lymphocytes, and also some ongoing analysis on fecal microbiome. First of all, we will have to wait some time to have a mature follow-up in terms of overall survival. At the data cutoff of the 1st of August, only the 28% of survival events were recorded, so that we need definitely and luckily more time to have these data available. And I think that having a signal of benefit for the experimental arm also in terms of survival would strongly support continuing these efforts towards new evidence in the first-line treatment of metastatic colorectal cancer with the addition of immunotherapy. And we will have to wait the results of the translational analysis to have some more results in terms of potential predictors of benefit from checkpoint inhibitors in the microsatellite stable subgroup. I think that based on these results, having new trials designed with this aim may be a good idea to improve the treatment of metastatic colorectal cancer patients. The AtezoTRIBE study is an independent, no-profit trial that was conducted in Italy, mainly thanks to the enthusiasm and the commitment of a lot of investigators throughout Italy. And I think that their enthusiasm and their interest in improving the outcome of our patients should be recognized as a very important starting point to have these successful trials and these good results to improve the treatment of this disease. So I'd like to thank all of them. I'd like to thank Roche for having partially supported the study, and all of the colleagues who contributed to this clinical effort, also in these hard times of the COVID pandemic.